Okay, let's get started. I'm really delighted to have Professor Brian Beliveau join us in the Spatial Omics seminar series today and to talk about the oligo-based imaging of nucleic acids in single cells. And I think Brian got his BS MS combined degree from Johns Hopkins University. And afterwards, he moved to Boston to study in Department of Genetics and Harvard with Ting Wu. And after his PhD, he stayed in Boston and working with Peng Ying in the Waste Institute. I think during that time, he did excellent, terrific work in the spatial omics and the spatial imaging and develop the Oligopaint. I guess Brian will talk about that today. And I think in 2018, Brian moved to the West Coast, University of Washington, to be an assistant professor there. So I'm very glad to have you, Brian, and we look forward to your talk. I think probably I need to allow you to share the screen. Sorry about that.
Okay. Well, I'll just say as you do that, thanks so much for the intro. It was very kind, and for the opportunity to speak and present our work. Let me see if I can go ahead and get those slides up.
Yeah, it should work now.
Okay. Cool. So can someone confirm that you're seeing my slides since I can't see anymore?
Yes.
Fantastic. And this is my first rodeo giving a Zoom seminar, so I hope you all bear with me. I've done a little bit of teaching. That was a little while ago, earlier in the pandemic. So do you see my mouse when it moves or no?
Can I try it again? Seems like no.
No, it seems like no. What about if I do this?
Yes. Yeah, this works, but if you go to full screen, I think there's a function you can choose the laser pointer.
Interesting.
Yeah, if you. Oh, you're using Keynote.
I'm using Keynote, but now can you see my mouse?
Now we can see.
Okay,
yes.
Well that's good enough, I guess.
Okay.
Well, thanks for your patience, and I'll just go ahead and get going then. I'm just gonna quickly move my Zoom window out of the way so I can see my title. Okay, great. Yes, I'm an assistant professor at UW Department of Genome Sciences. I've been there for a little, getting close to 2 years now. And so today I'll go through some of the technologies I've been involved in developing throughout the course of my training, and a little bit about what my lab is doing now that hopefully will touch upon some areas that are of interest to the audience. All right. So I guess what's going to happen is I'm going to hit play and then my mouse is going to go away. So we'll just see how it goes and we can toggle back as needed. All right. So biologically, my principal interest in life, scientifically at least, are chromosomes. I really love chromosomes, and I think, as the name suggests, the name itself comes from the visual. It's a description of looking at something visual. It means staining body, and I just think they're so beautiful to look at. And if I had my choice, I'd always prefer to look at chromosomes that look like the image I'm showing you on the left of your screen, where you see these beautiful textbook kind of condensed mitotic chromosomes. And if we wanted to learn something about the genome in terms of how the linear DNA is organized into chromosomes, or how many chromosomes there might be, etc., it's reasonably straightforward to do it from this kind of image. But of course, in biology, this is an ideal case, because if we were to say, for instance, take something like an asynchronous culture of cells growing in TC and fix them and stain them with something like an intercalating dye, like the DAPI image I'm showing you here, we see a much different picture like you're seeing on the right. So this is this kind of smeary punctate mess that is the interphase nucleus. And this is, for us, a kind of a principal biological motivation to make the point that this interphase nucleus structure is very complicated. And this is really what anchors a lot of the technology developed and the biology that we think about investigating. And I really think the scale of this problem is hard to conceptualize and to illustrate or to try to visualize. So every few months or so, as I get ready to give new talks, I go on Google image search and I try to find things that show like disorder or clutter or chaos or disarray, just to try to get, something tangible to lead on as an image for how this compartment might look in this organization. So the picture you're seeing here is a cabling disaster. I've used other ones in the past. I'm always looking to crowdsource this. Like we've tried messy dorm room, we've tried this and that. So if you have any suggestions for what might do a better job with this analogy, feel free to hit me up at any of the contact info at the bottom of the slide. And I think in part it's pretty important to think about why this organization is interested in studying, why it matters when we start to think about, well, what's in this big nuclear soup? So we know that we have, for instance, 10 linear meters of DNA, if it were relaxed, that has to fit into a compartment whose diameter is a few microns. We have tons of proteins and RNAs and lipids and sugars in there as well. And then if we think about the genetic information that's encoded in the DNA, we have lots of genes, we have many regulatory elements that can help drive expression. We're making lots of different transcripts, different transcript isoforms. And this all has to go on in a compartment where we have to regulate spatial access to DNA for things like transcription and repair and replication. So I think there's a lot to kind of think about and still try to unpack in terms of understanding how the nuclear compartment functions in biology. We know that my kind of tongue-in-cheek examples from the last couple of slides in terms of these images aren't really accurate in that the nucleus itself is a highly ordered compartment. It's not the cabling disaster or the bowl of spaghetti per se. And a clear example of this, for instance, would be the tendency of individual chromosomes to occupy discrete territories in nuclear space. Another concept that we have begun to understand through work by many labs over the last couple of decades is this idea of functional partitioning of the nuclear territory, meaning that generally, for instance, the nuclear interior is more associated with the transcriptional activity and transcriptional activation, and the periphery is more associated with things like gene silencing. So I think ultimately it's going to be a combination of spatial mapping in terms of understanding where individual aspects of the genome are in a given cell state or a cell time, combined with understanding how the microenvironment is affecting the biology of the sequences for us to get a really integrated view of how genome organization impacts the processes that happen in the nucleus. But to transition a little bit more into what I'll talk about for the rest of the time, our lab, and as a trainee in some of the labs that I was mentioning in the introduction, we've focused on trying to develop new tools that are easy to use and accessible to help ask questions, primarily in this space, but I think there's also nice applications, especially for things like spatial transcriptomics with the SABER technique I'll talk about in a little bit. So the first technique I'll introduce that's kind of the bedrock of everything else is this Oligopaint technology I developed as a grad student in collaboration with Eric Joyce when I was in Ting Wu's lab. And then since then, in part in Ting's lab and then moving on to Peng's lab for my postdoc, we started to apply this kind of painting technology to do super resolution imaging with a technique called DNA paint. I'll talk a little bit about that. And then most recently, when I was a postdoc in Peng's lab, and this is something we're using a lot now in my lab, we developed a technology that some of you that would have attended CNM's talk a couple weeks ago would have already heard about, called SABER, for in situ signal amplification with multiplexing. So to lay the foundation, I'll first give you a little bit of information about what Oligopaints are and how they work. And again, this was work that was done with Eric Joyce when we were both trainees in Ting Wu's lab. So Oligopaints is a somewhat new way now to do a more classic technique of FISH, or fluorescence in situ hybridization. So the FISH itself derives from ISH, or in situ hybridization, and this was first reported in the late 1960s by Mary-Lou Pardue and Joe Gall. And since then, it's been kind of updated and modernized a little bit. So one of the key differences, for instance, is adding the F, and instead of using radioactivity to detect the signal of specific chromosomal regions or RNAs, we can now use fluorescence. And this is great because it's a lot safer and a lot faster and more sensitive. The general, the zoomed out view of what this technique does is that we target cellular RNA or DNA using the inherent complementarity from base pairing using nucleic acid probes. And by virtue of how this works on a microscope, this is fundamentally a single cell technique, which is great. So I imagine this audience is quite familiar with all of this, but just to remind myself before I go through the next few slides really too quickly, the basics of how a FISH assay would work is that you'd have a fixed sample. So unfortunately, everything that we do for this is dead. I always get the question, can we do this in live cells? And I think it'd be awesome, but at least for now, we're restricted to these fixed samples, typically with formaldehyde. We can then denature the genomic DNA for targeting DNA. For doing RNA, we don't often have to do that step, although there may be secondary structure we need to disrupt. And once we've done that using a combination of heat and chemicals if necessary, we hybridize an excess of probe that's complementary to the sequence you want to target. And this probe bears a label that we can in some way detect using a fluorescent microscope. So the oligovein approach does this a little bit differently than previous iterations of the FISH method. And the principal difference really comes from the source of material for the probe. So a classical FISH experiment would involve either cloning out a region of the genome into a vector like a BAC or a plasmid or a cosmid, and amplifying that material in vitro before processing it and fragmenting it such that you have a pool of labeled fragments you can apply to try to do the hybridization. More recently, you can do this using PCR products. So that's a nice way to kind of streamline that process and make it a little bit more specific. But in general, in those cases, you're still using relatively large chunks of genomic sequence that you pick in blocks. And you're starting directly from the source material of the organism's genomic DNA or cDNA. So by contrast, the oligovein technology and a couple of companion technologies that were introduced by Agilent and Wendy Bickmore's lab around the same time work a bit differently, and they use synthetic DNA made by massively parallel oligonucleotide synthesis as the starting source of material. So we'd call these kind of a complex oligo library as a term. I mean, generally what these tend to be are microarrays. So the same technology that used to be used to look at gene expression changes and used for array CGH, only instead of keeping the array of oligos on the solid substrate where they are synthesized, there's a step that cleaves them off and pops that pool of thousands to tens of thousands, maybe even millions of sequences into solution. And the practical things to know if you're working with it, you're thinking about maybe using this technology, you haven't used it before, in terms of what it costs to get set up with one of these libraries. It depends a little bit on the scale and the complexity. So for this slide actually is a little bit out of date. So the way that you would do it if you want a lot of material for each individual oligo typically would be to order individual column synthesis products from IDT in like plate format. So similar to what you do for ordering tubes of primers, here you're getting on the order of nanomoles per sequence. It's a lot of material, but you're paying a lot more because of it. And this doesn't scale well beyond a couple hundred oligos. IDT has a new product called an OPOOL, which is kind of a nice intermediate between what I just described and the more complex libraries I'm about to describe. So I think, for instance, if you're interested in targeting a couple of genes for a SABER FISH experiment, jumping ahead a little bit, we found that that's a nice intermediate point where you're paying like $100 or so to get about 50 or 100 experiments worth of material, and you don't have to do as much work on it. The other options scale much better in terms of the number of oligos you can get per dollar. And so these are the array synthesis products I was mentioning. You can get anywhere from a few thousand oligos for around $1,000 up to, you can push if you want beyond even 100,000 oligos. There are a few different companies now that'll sell you pools in this format. So the larger you make the pool, the cheaper in theory you're getting. The deal you're getting in terms of base pairs per dollar gets better. The fundamental difference between the 96 plate oligo that I have italicized and all the other ones here is that the amount of material you're getting from these array synthesis products that are cleaved into solution is very, very low. So you're getting less than like attomoles or something like that per species. So in order to be able to turn that into enough usable material to actually do an experiment on a TC sample or a tissue section, you need to do some molecular biology. And that's where we started with when we were developing Oligopaints about a decade ago, was kind of figuring out a process by which we could, for instance, put primer pairs around these sequences that are going to target the FISH probes and to efficiently make these in bulk to the scale of like picomoles or so, which is what you need to do a FISH assay. And then we can apply them to try to target, a given stretch of chromosome or messenger RNA. And the good news is that this works really well. So we can do things, for instance, like quantifying our efficiency of labeling chromosomal DNA at targets ranging from 10 kilobases up to multiple megabases. And we see generally on the order of 90% or more of cells displaying 1 or 2 foci, which is great. So that tells us that we're doing a very good job of interrogating the DNA that we're trying to target in our sample. And these data themselves are quite old. So we and others have made a lot of updates to the molecular biology and FISH protocols aspect of this. So I think right now it's fair to say that this is a pretty workhorse and routine technique that's being used by a lot of groups. So it's really, satisfying as a developer who's involved heavily in making this technology way back when to see what it's out and doing in the world. I think this slide, besides just showing one of my favorite chromosome images that was taken by Eric Joyce, I think does a nice example, provides a nice example of what you can get from using this kind of technology compared to the more standard fish approaches. So what I'm showing you here are salivary polytene chromosomes from Drosophila. So this ends up being 20 megabases or so on chromosome 2R, and that's about a fifth of the fly genome, at least the non-repetitive content. And we're targeting it, in this case, with a library of 180,000 oligos, making a 3-color banding pattern. And just trying to do this any other way, I just don't think would really work. I mean, you could somehow get the backs or the conventional fish probes you would need to make labeling happen in this kind of way, and did it yourself, you can maybe do this for $100, assuming you could source and raise all of those. This is just really factoring in the cost of the probe material itself and the molecular biology to make the fish probe. You could buy it maybe for $3 grand. And in this case, once we had the libraries, and that was of course an upfront investment, we were able to do this for less than $3. So I mean, the scaling of this technique is really powerful. I think you'll see that come out in some of the later stuff I present. And yeah, I think the other really nice thing is that the scaling works not just in terms of the cost effectiveness of the molecular biology once you get going with it, but also just in terms of the general protocols. So the FISH protocol we used here is essentially identical to the FISH protocol I showed you on the previous slide, targeting small and big regions. It's pretty much all the same. You'll have to tweak it, of course, for tissue sections and things like that. They may have nuances, but the general, when you're working in a given system, the complexity of the library doesn't really seem to alter too much how you have to think about the experiment. So I think that makes it pretty straightforward for investigating a lot of different types of questions. One of the really cool things about the technique too is it's really open to any system that has a sequenced genome. So this slide's also a little bit out of date. I'm sure there are more organisms I could put on here, but I think this is all the same a pretty nice illustration of the breadth of what people have been able to do using this oligo-based FISH approach. If you have a sequenced genome that's reasonably good, you can go in and use the informatic tools we use to design the probes, and I'll talk about those a little bit later, and go ahead, including even in things like mammoths, they're doing it, which is kind of crazy. So this has also been really fun to see as a developer. And I'll leave you here with the Oligopaints story since it's been out for a little while, just again to highlight some aspects of what I think makes the system powerful that'll illustrate how we're able to harness it to do some new things in the remaining sections of the talk. So the way that Oligopaint probes generally work is that we have a section of homology to the target, be it the genomic DNA or the RNA, that is typically on the order of 25, 30, 40, 50 bases maybe. Depends a little bit on the conditions you want to use experimentally. And by virtue of how we make these with molecular biology, these sequences, or these probes rather, often have an overhang sequence on their 5' end that comes from the PCR priming step we need to do. And you can potentially put a fluorophore right there. So it's a pretty effective way. For instance, you can buy one fluorophore labeled primer and do PCR and apply it to many, many different oligo species instead of having to order 180,000, say, fluorophore labeled oligos, which would be super expensive. You can order one and amplify the whole library and get it onto all of them. And then you also potentially can encode overhangs deliberately into your oligos as well. So for instance, you can make a binding site for what we call sometimes a secondary oligo that works very analogously to how a secondary antibody would work. And our ability to control at a base pair level, the full sequence of this whole, of each probe in our library really is what's opened up, I think, a lot of cool engineering avenues in terms of pushing what FISH technology can do. So I'm highlighting a few of these here. This is work from us and others that have done all of this. One set of work from Ting's lab when I was there was to develop allele-specific DNA fish that scaled to non-repetitive regions. So here we can target a pair of probes that are differentially labeled with single nucleotide information when we have phased variants along the different haplotypes and get allele-specific labeling, which is pretty fun. And then I think a bit more relevant for this audience are these ideas of doing these massively multiplex serial imaging experiments, either the chromosome walking style experiment. So here's an example from Jiawei Zhang's lab doing that, or the spatial transcriptomics, which I think is kind of the, probably the reason I'm being invited here is for this kind of stuff. And there's obviously a lot of exciting methods in the space. I'm just showing two of the earlier examples with the MERFISH and the seqFISH techniques. So I'm going to transition for a few slides and just give you a quick taste of some of the super resolution that's possible. I don't know if this intersects as well with the spatial omics, but it is an exciting area. So just to give you a bit of a background into what's possible with these techniques, I'm going to go through a quick example that in some work that was done in collaboration with Hiroshi Sasaki, who's a postdoc in Peng's lab, as well as Ralph Youngman's lab in Munich. So when we talk about super resolution imaging, the problem we're essentially trying to get around is the property of diffraction limiting how small of an object we can resolve with our microscope, no matter how much money we spend trying to make it fancy. So practically for the wavelengths that we are most likely to image in an in situ hybridization experiment, say, that ends up bounding you on the order of about 200 nanometers or so in change in the XY or lateral dimension, and 500 nanometers or so in the Z or axial dimension. And I think the way that this this concept is often introduced is this idea of two objects being close to one another and whether or not they're resolvable. And that's a very useful way to anchor understanding around this topic. But there's also, I think, another more practical way to think about it that I like to introduce, which is just thinking about volumetrically, what does this mean? So for instance, if I do FISH targeting on genomic DNA, targeting a very small region, because of diffraction, we know that there's going to be a characteristic minimum size of what I see on the microscope. My signal can only get so fine before it essentially becomes the same size. So what does that practically mean in terms of these imaging experiments? So if you think about what would be bounded by this, you could, for instance, take relaxed B-form DNA and stretch it out, and that would get you about 600 bases across in the lateral dimension and maybe 1.5 kb in the axial dimension. But of course, in the context of a 3D genome in the nucleus, we don't really think of naked relaxed B-form DNA. So if we do a thought experiment and some back of the envelope calculations thinking about chromatinized templates, for instance, I think you could in theory fit somewhere on the order of 15,000 nucleosomes, which would be a couple megabases of compacted DNA into that volume. Let's say in practice, that's probably not likely, having imaged a lot of fish spots in my career. So I think anything above a couple hundred kilobases is likely going to give you a larger than diffraction size limited spot. But what this is telling you is that, and many proteins can fit there too, a lot of the length scales that are really relevant for biology that people are excited to understand, like enhancer promoter interactions and localized folding and gene bodies and things like that, like that's going to be completely invisible no matter how much money you spend on your high numerical aperture objective or whatever else you can do to try to make your light collection as good as possible. The diffraction is just fundamentally bottlenecking things at this length scale of hundreds of kilobases or so in terms of our ability to resolve information. So there are multiple ways around that problem, including just doing some clever tricks with diffraction limited imaging that I don't have time to talk about today. But the other avenue is to use some kind of super resolution microscopy that, as the name would suggest, just attempts to get around or below that diffraction barrier in some way. There's a lot of different kinds of super resolution microscopy, which I wouldn't have time to go through all of, although I'm happy, in the questions if people have questions, to try to answer on that front. I'm going to talk specifically over the next few slides about a certain type of super resolution microscopy called single molecule localization microscopy, or SMLM. So the way that SLMM works is that you would take an object that you're normally labeling with a population of fluorophores, because we always need at least a couple dozen of them or so to see anything in the nucleus over background, or anything, maybe on the RNA and the cytoplasm, a bit fewer, but you're typically talking at least a population of 10 or more. So instead of having these all fluoresce as a group, the idea is that you somehow build stochasticity into the system, so individual dye molecules blink on and off at different times, such that you can then separate in space and time, if you take a time-lapse image of a static object, these blinking events. And then if we're pretty confident that we've captured an individual molecule blinking as opposed to a population of fluorescing in a diffraction-limited volume when we see it, we can make an estimate of the point source. So we're not really resolving the molecule any finer, but we're making a mathematical estimation that's essentially a statistical calculation. And we can do that with a precision that is about twofold better than the diffraction limit. And what we then do is reconstruct an image that ultimately comes from taking a 5 or 10 or maybe 30-minute time-lapse image, again, of a static object that's made up of thousands or more of these individual localization estimates. And the flavor of single molecule localization microscopy that I'm going to talk about today is one called DNA Paint. So this is a technology that Ralph Youngman developed at the end of his PhD days in Fritz Simmel's lab, and he went to Peng's lab, where I also postdocs as a postdoc there, and really fleshed it out and has continued to do really awesome things with it in his own independent position at Munich over the last few years. And really the important thing to take home from DNA Paint that hopefully dovetails somewhat well with the rest of the talk is it's another DNA hybridization powered technology that comes from placing specific sequences of DNA in certain places and programming binding events to them. So in this case, in the illustration I have here that I hope you can follow, this is where, actually, we're just going to do it live. I'm going to use my mouse here. I just think it's going to be a little bit easier, and I'll go back to the full screen. So this is, say, this blue here is our genomic target we're trying to hybridize to. Let's say this is a messenger RNA, and here are our complementary DNA FISH probes. What we do is we put a short overhang of sequence onto each probe that's shared for the probe set. And these are typically 9 or 10 bases long. And we do the imaging in the presence of the complementary sequence that has a fluorophore labeled. So just by random collisions, you'll get transient hybridization and binding of the fluorescent oligo onto these sites. And this will last for about a second or so in the way we've programmed it, then it'll just spontaneously dissociate. So in the diffraction limited field where this target is located, if you look at the intensity trace in time, you basically get these blinking events where it blinks on, it stays, it dissociates, and then this happens again and again and again. And this is what allows us to get single molecule blinks that we can then super resolve. And nice ways that we can kind of validate that this approach works is, for instance, programming two-dimensional DNA and origami nanostructures, putting them down on a surface, like I'm showing you here on the top right of the slide, and then trying to resolve these strands that we projected and define patterns that are certain distances apart. I find this very satisfying because if we say we have X nanometer resolution in our imaging and we actually have features that are on that length scale or smaller that we can resolve, it really does, a convincing job of saying that that resolution estimate that we make has meaning in the system. The other really powerful thing about this DNA paint technology, other than the programmability in terms of getting that on-off blinking rate right, which is pretty important technically for getting good image quality, is our ability to multiplex this. And this is a concept that's going to come back up in the SABER section that I'm going to go to a bit later. So because the blinking is driven by sequence binding sequence, we potentially can design multiple orthogonal sequence pairs to use in multiplexed imaging. So the way that we can do this is, for instance, in this example, there are 4 different flavors of this docking strand, as we call it, that's going to target the paint imaging arrayed along, say, a stretch of chromosomal DNA. So the first one you're seeing here is the magenta sequence, and we can image that just as I described. And then because the binding is so transient, we can essentially just do a simple buffer exchange and then add the sequence that's complementary to the next target, which in this case is green. And the key thing here is that the fluorochrome is actually the same. So we're using the same imaging channel. We're just doing it in serial rounds. And for super resolution, that has a lot of advantages in terms of helping to account for things like, or avoiding the need to account for things like chromatic aberration that can be quite tricky over these length scales. And we can essentially do this n times. As long as we can continue to design sequences that we predict will not interact with each other in the experimental conditions, we can have unlimited colors, so to speak, in this way. And here's a nice example of that being implemented where they designed 10 different digit DNA origami nanostructures that were all present in the same field of view, and then imaged them in 10 sequential rounds, where in each round of imaging, one of the digits was being visualized. So I'm going to show you just a little bit of data from using this to actually look at genome organization, which is a harder problem than looking at these origami nanostructures. So what we've done here is design an Oligopaint library that targets this 15 megabase or so region of the human X chromosome. The specifics of how the targeting happens is not so important for this, but for what it's worth, the library design was in part inspired by a data set with topologically associating domain calls from Bing Ren's lab that Jesse Dixon published a long time ago, the original set, that is, the 2012 paper. But I think more importantly for the purposes of the next couple slides, this is really just to illustrate the power of what this kind of imaging potentially can do in a multiplex context. So we went ahead and used the same multiplex approach to do 6-color imaging on, in this case, human tissue culture cells. So this is what the diffraction limited version of the imaging would look like. I have the labels down below at the bottom of the slide, and the number you see for nanometers is the focal height of the signal. So we just took a single 2D plane of a 3D object, where in this case, we were able to get the bulk, but not all of the signal. And as we moved down the chromosome, this actually moved a bit in 3D space. And then this is what the super resolved version of that would look like. So we get a little bit of both alleles, but the one in the center is the one that we really focused on. The other one's a bit out of focus. So to kind of show you what we can get out of this, of course, you have to take it and make it bigger. So this is just that focus in the middle. And then if we zoom in and look at the 6 color, this is what you see. So I personally never get sick of looking at these. As some of you may have noticed, this actually is my Zoom background I'm talking in front of right now. It comes from this image. And I think what's so striking to me, and we've also seen this now in work that's come out of Ting's lab more recently, and work from Zhao's lab and from Alistair Becker's lab at Stanford, when you look at these multiplex storm or DNA paint images, the way that the chromosomes just interdigitate, and you can see so much going on in terms of the signal nestling around different colors and then packaging together. It tells us that we have I think a technology that is still pretty nascent, but might really be aimed well at trying to ask pretty nuanced questions about how different genomic regions are intermingling and associating on these very fine length scales. To put a little bit more context into that, here are some of the quantitative numbers we're able to extract from this imaging. This is just a simple summary we can get. There's many more advanced ways to analyze these data, but just as a zoomed out view, we can look at, for instance, what our effective resolution is. And in this experiment, we are getting on the order of 20 to 30 nanometers in the lateral dimension. With different optics, you can probably cut that in half. But with the optics we use, those are the numbers we got. And this kind of approach in general, I think, is going to help ask questions along the lines of, like, for instance, if you define 2 domains, either topologically associated domains or other just intervals of genomic DNA that you think have interesting features based on, say, a sequencing experiment or a high-C experiment, you can ask questions about intermixing between those adjacent domains. I think one of the pressing questions that we still don't have a great answer for is whether there's stereotypical conformations for a given set of sequence, like a domain. From my experience, I've described these as snowflakes. So you can image the same region 100 times and they all look different. But there may still be, that said, commonalities in that repeat between different sequences. So I think we're still a little bit early in getting a sense for that. But I think these kind of approaches with the resolution they provide will be great tools to look at that heterogeneity in terms of the structure. And I think one of the big ones that you're starting to see more of come out, including work that just came out from Stephen Wong's lab, is looking at correlations between structure and gene expression state. And this is, I think, going to be the next wave, and in some ways why this section could still connect reasonably well to the spatial omics world. But making that connection stronger, I think I'm going to now transition for the bulk of the rest of the time to talk about SABER FISH, which is one half of the SABER story. I know you've already heard about ImmunoSABER from CNM a couple weeks ago, and I'll just touch on that briefly at the end of this section. So this is a story that happened in Peng Yin's lab when I was there as a postdoc, as well as Connie Sepko's lab. And this was a great collaborative effort led by Josie Kishi, Sylvan Lappin, and Emma West and myself under the directions of Connie and Peng. So let me tell you about SABER and how it works. SABER is one of my favorite things. And in order to explain the SABER acronym, for those of you that haven't seen Sidam's talk already, I have to throw another acronym at you first, so I apologize for that. But I do think this is some really cool molecular biology, and I never get sick of going through it, so I'm going to take you through it now. There's a reaction called the primer exchange reaction, or PER, that Josie developed based on some work that was going on in Peng's lab a couple of years ago as a way to synthesize DNA in defined ways. So Peng's lab is getting into the cellular imaging world more and more these days, but historically it has stronger connections to DNA nanotechnology, and still does. So one of the things that lab likes to think a lot about are things like DNA computing, and can you use biological molecules to do things like encode state transitions. So there's a simple example of that shown on the right side of the screen here. This might be a good slide to bring my mouse back, actually, so let's do that. So say, for instance, we have what we're going to call a primer molecule, which is species A. So in this case, this is a 9 or 10 base single-stranded piece of DNA, very similar to a PCR primer, but a bit shorter, that does have that 3' hydroxyl that's needed to continue to extend the sequence further, potentially, if it sees a polymerase. And we're going to put that into solution with this hairpin here. And I'm just going to explain quickly the schema we use to describe some of these sequence domains in this kind of format. So if we have a sequence A, then A star would refer to the reverse complement of that sequence, such that they would base pair together if they were to meet under favorable conditions. So following along that, we have a B star stem loop here in our hairpin where these sequences are base pairing together. And the goal of PER is to have this special hairpin, and I'll explain a little bit more how it works in a sec, be able to sense that it's seeing this A molecule and affect a change, which in this case is to append the B sequence onto A. So this is a little computing thing we could kind of look at, where if the right conditions come together, we can make a record of it by making a new sequence, which they're calling a transcript, as a result of that coming together. So I'm going to explain how this happens first, and then I'll explain why in the world you should care about that with regards to spatial omics or cellular imaging on the next few slides. So the first thing that happens in the cycle, as you may have guessed from the way I introduced things, is that base pairing needs to happen. So this A sequence, just by transient collision, is going to hybridize to its complementary A star sequence on the hairpin. So once these two molecules come together, if this happens in the presence, I guess I have to do this, of a strand displacing polymerase, you can get extension across the stem loop until it hits that double black dot with a little bar in between. This is because there's a stopper sequence put there. There's a few different ways you can affect this stop. The details aren't super important, so I'm happy to answer questions about that, but for now, I'm gonna skip over the minute detail there. And just suffice it to say, what's important is that there needs to be a roadblock placed there that stops polymerase from synthesizing any further so it doesn't blow around the stem loop. If that happens, if those conditions are achieved, then what you're actually left with is an equilibrium state, because the flap that's extended back has the same thermodynamic strength binding to the B star sequence as the newly synthesized B sequence does, so that those two molecules are in equilibrium fighting for the ability to bind B star, the new B and the old B, but through the process of branch migration, they're just pushing each other back and forth. So the key thing that happens is when you get to the state that's in the center bottom of the slide, where the new B is pushed back. Because similar to DNA paint, we can program the kinetics and thermodynamics of these interactions. We deliberately make the AA star pairing quite transient. So just by the spontaneous dissociation, this AB chimera that we've just made will just diffuse right off and float away. And we've done one synthesis cycle at this point. So the B hairpin's free to initiate another synthesis cycle somewhere else, and we've now added the B domain onto A. Okay, so that's a PER reaction. So what does that have to do with anything with regards to imaging in cells? Oh, actually, sorry, I'm getting ahead of myself. We have a really beautiful animation that Leijin at the Wyss Institute made to kind of show this process. So let's see if that'll play. Great. So everything's flipped upside down. I apologize for that, but it's the same process other way. So again, the idea is that the primer can bind its complementary sequence on the hairpin. You bring a strand displacing polymerase. You synthesize until you hit the stop. And now we're at the point where you're going to have an equilibrium state with branch migration happening between the new and old sequence that's on top of the template strand. And eventually by branch migration, you'll push that new B back and you'll get dissociation. And then you potentially can do this again and again and again in a programmed fashion. So you can make transcripts with defined orders based on the hairpins that are present in the solution, which is kind of the point of the initial PER paper that started all of this work. Okay, great. But jumping ahead now, to the cellular case, this becomes really powerful for us when you encode A star in the place of where B star was on the previous slide. So you basically copy the same sequence onto itself. And so what happens when you do this is that every time you make an extension, the molecule you've made is then capable of being extended again because it has the right sequence to bind the relevant part of the hairpin to allow it to be extended, right? So what this effectively does is concatenarize the molecule you're extending, making many, many copies of a short sequence that's repeated over and over and over again. So this is a really efficient and cheap reaction to generate potentially kilobases worth of repetitive DNA, the same 9 or 10 bases are just repeated over and over and over again. This is very similar to a PCR to set up. It costs about $1 per reaction. It's isothermal at 37. You run it for 1, 3, 4 hours. And I think the beauty of it is that this repetitive DNA is just a nightmare to get any other way. The only other way you can kind of get this type of thing is by chemical ligation. If you ask an oligo company to make this, they'll probably just laugh at you and or just get really sad because synthesizing these things on a column with their repetitive nature over the length scales we're talking about is just a nightmare, and no one's going to want to do it. So what Josie really has done by introducing PER is make a really nice framework in a molecular biology sense, to synthesize these very difficult molecules very easily. And here's a quick animation showing you what happens if you add the same sequence to itself over and over. So instead of adding multiple different color domains, now we're just getting one long unicolor domain because it's the same sequence over and over. Great. Okay, so how this connects back to the omics world is that, we're looking at this PER reaction as Josie was finishing this development and thinking about the kind of things it might be useful for in a general sense, not even necessarily just in the cellular context. And it became very clear looking at it that we had something on our hands that looked very similar molecularly to some of the other technologies that are being used very actively to amplify signals in cells and tissues, like the RCA technique from David Ward's lab and the HCR technique from Niles Pierce's lab. So we thought about it a little bit and realized that we think the PER reaction certainly brings some interesting stuff to the mix, in that it makes single-stranded DNA. By the virtue of how we make it, it has very, very low secondary structure, which we think is important for hybridization kinetics. And it's just cheap and easy. Like it's one of the easiest reactions I've ever done. So we're kind of curious to explore whether we could use this in a similar context to the RCA and the HCR approaches. And so conceptually, what we're thinking about essentially is making something quite similar to the RNAscope product you can buy, but a little bit more on the order of providing a toolkit for researchers to do something in their own hands cheaply, as opposed to making a product that someone would have to buy pre-synthesized at a reasonably high price. And that is the genesis of SABER, or signal amplification by exchange reaction, or by PER, which is a term that Josie coined that I think is quite clever. And kind of the joke here is we're taking oligos and extending them to make them bright. It's kind of like illuminating a lightsaber. So practically what we're doing is we're extending either probe oligos themselves or the secondary oligos I described earlier in the talk that can bind to probes that are docked at the sites of hybridization by 500, 600, 700 bases, maybe a bit over a kilobase, depends a little bit on what you're doing. And this in practice creates somewhere in the order of a few dozen to maybe several dozen binding sites for a labeled complement of what you've extended. So we normally extend 10 base repeats, and we typically add a 20-mer, so a dimer of the repeat, reverse complement label with a fluorophore. And effectively, this is producing somewhere in the order of like 10 to 40 folds signal amplification. So the way that SABER works, I do think makes it a little bit distinct from the RCA and HCR approaches, is that we first do the synthesis in vitro. And this is really key because it lets you QC what you're doing before you go on to do anything more complicated or expensive. I think for me, I love those techniques. I think they're tremendously clever and really powerful, but some of my frustrations from using them over the years is that if I get an experiment where I don't see my desired result from it, where I need a reaction to be happening in situ, be it an assembly reaction for HCR or an enzymatic reaction for RCA, it's just hard to know what to troubleshoot or what went wrong. So by decoupling the steps and doing the synthesis in vitro, where you can QC it and kind of add a known commodity first, I do think that makes SABER quite powerful in terms of being an easy to deploy technique for this kind of thing. So once you do your PER, you can purify it. You don't have to, it depends what you're doing. It's a relatively straightforward couple-hour reaction either way. You essentially can then just go ahead and do your in situ hybridization as normal. So you can hybridize PER extended fish probe oligos, Oligopaints, using the same exact protocol as you would normally. You don't have to change anything. They seem to work fine as long as you don't make them too long. Or you can, again, hybridize probes that provide secondary oligo binding sites or bridge sites or readout sites. There's many different terms for this, but these single-strand overhangs that can then be used to recruit a common sequence that's been extended in a separate PER reaction. And then that's your standard protocol. So what you're adding here is just one additional step, which is a relatively quick hybridization. This can be anywhere on the order of 5-ish minutes in the best case, maybe to a couple hours in a thicker tissue context, where you add this fluorescent oligo that's going to bind the concatenamers and blow up the signal. So we went ahead and validated this as part of introducing the technique. Of course, for DNA fish, we went to my favorite target, the metaphase chromosomes, where you can use the linear order of what you see and the type of chromosome you see it on to get great confidence that you're seeing what you expect. So here we ended up targeting about 4 megabases or so on human chromosome 1, and we get the expected co-localization pattern of these 3 colors. And we of course can also visualize this in interface cells too. For RNA fish, the validation is a little bit different. So the typical way to do this, and this is taking a play from the Arjun Raj playbook, is to take your transcript and kind of break the probes into 2 flavors. So in this case, 2 different PER sequences being extended and being recognized with different complementary oligos bearing distinct fluorophores. So practically what we're doing is we're taking, in this case, the CBX5 RNA in mouse embryonic fibroblast cells, and putting 61 green probes and 61 magenta probes on, imaging them separately, and then asking for how concordant the two images are after automated image analysis and segmentation. And the answer we get is phenomenal. Like we're getting 92, 95% overlap of the puncta, depending on which one we use at the reference channel. And these numbers are absolutely stellar for this kind of assay. I think the rate limiting step ends up being the segmentation itself. So we feel very good that the SABER approach is maintaining the inherent fidelity of either of our June single molecule FISH technique or of our Oligopaint technique when we go to use the SABER on top of it. And I think, honestly, the real power of SABER in some ways comes from its multiplexing and not its amplification, but they're related. So I'm going to explain that quickly here as I finish up. So what we can do with SABER in a very similar way to what we were able to do with the DNA paint is program distinct sequences to concatenarize in the PDR reaction and then recognize with the fluorescent detection oligos. So here's an example of that where we have 7 different RNA targets, and we're going to add 2 protein targets at the very last step. So what we've done, and this is in mouse retinal tissue, work by Sullivan Lappin and Emma West in Connie's lab, who did all of the sample prep and imaging here, is they've hybridized all 7 RNA probes in one step overnight, and then they've added in a secondary hybridization the fluorescent oligos for the first 3 targets in 3 distinct fluorescent channels. So we go ahead, we do that hybridization, the secondary hybridization that is, we wash, we go put on the confocal and we image, and then we can just shift the solution conditions by introducing formamide. And because these are only 20mers that, in this case, actually have an elevated AT content, they are very rapidly displaced under conditions that do not at all disrupt the base pairing between the FISH probe and the RNA or DNA target. And that's one of the powers of this technique. And then we can, 5 minutes or so, those are completely gone. We wash again, and then we can go ahead and add the same 3 fluorescent dyes, but now conjugated to 3 distinct sequences to bind the next 3 targets, and then image those. And they do this again and again and again. So, in theory, we can do unlimited numbers of serial imaging rounds as long as we don't run out of these PER sequences to use. Right now, we have about 50 of them, and there are ways we potentially could recycle them as needed. So I think there's a lot of potential for doing many, many colors using this approach, even without using a combinatoric code to boost the number of colors you can get per round. And the reason that this works so well has to do with the kinetics of DNA hybridization. So for any of these sequential imaging techniques that rely on using readouts that vary by round, so you hybridize to target 1, then you somehow remove that signal, and then the next round you hybridize to target 2, and so on and so on. The rate limiting step is always going to be association of a new label with the target that's docked in situ. And that's just because by the collision frequency is really what's bounding the ability of these things to hybridize, and the concentration of what's associated either on the RNAs or on pieces of chromosomal DNA you're trying to target in situ is extremely low. And there's just no way to get around that bounding if you're trying to do a 1-to-1 stoichiometry between target site and labeled oligo. But SABER cheats because we make a whole big old repeat where there's many, many different copies of the same binding site repeated over and over. So we don't actually necessarily have to get to saturation. We just need to get enough oligo on there that we can see the thing. And we can do that much faster because we have that kinetic advantage. So I think this is on the order of like 5 or 10-fold faster than anything else you can do right now, just in terms of how long each association step takes. And then again, that is by far the rate limiting step in these multiplex serial approaches. So I think SABER, whether you want to use combinatorial readout like they do in MERFISH and SeqFISH, or whether you want to do something more conventional like 1 or a couple of transcripts around like the awesome FISH from Sten Lunderson's lab, this is an underlying molecular engine that one can take advantage of to push all of that through faster, no matter which one you want to go with. And coming back to the theme of the symposium, I think the power of this, given that it really seems to have a sweet spot for imaging, dozens of targets, something like that, in cell and tissue samples, is applying it to spatial transcriptomics. So to go from your single cell RNA-seq information, picking out 1 or a few marker genes for each cell type you're interested in that dropped out of the clustering based on differential or exclusive expression, and then doing this multiplexed FISH to reveal the patterns of these in the spatial context. So we did this in mouse retina using already known markers from Connie's lab, or we kind of expected to get the picture we're getting. But it's still very satisfying, all that said, to see it. And I think from a discovery angle, the SABER technology is fantastic and really well suited for applying the same kind of methodology where the underlying cell types and their distribution aren't as well known. So yeah, I'll just have one slide to plug ImmunoSaber quick as I'm finishing up. And you've heard about this. This is led by CNAM and you. So you can do this on proteins as well for people that didn't hear CNAM's talk. So you conjugate an oligo to the antibody you want to target the proteins with, and then you recognize that oligo you've docked onto the antibody with an extended complementary sequence. And at that point, it pretty much becomes the same as what I presented. So here's a nice example of doing multiplexed protein imaging in retina from CNAM and you and colleagues. And you can even potentially use now SABER to mark out different cell populations for sequencing experiments via fax. And this is work that Ryoji Awamoto did in Connie's lab, introduced relatively recently, called ProbeSeq. So if you're interested in SABER FISH or SABER in general, if you go to saber.fish, it's a great place to get started. We have a lot of info on the technology, the protocols you can use, how to design probes, and other things like that. My lab is interested, for instance, in doing chromosome walking, so making many spots along the chromosome and using the serial imaging to map conformations in 3D, which is seen nicely here in these GIFs. And I'm gonna skip through this slide because I think we're getting a little bit tight on time. And I'm gonna do about 3 more slides and then I'll stop and take questions. So I just wanna put a quick plug out there for how we actually get the probe sequences themselves, because this is a really important aspect of how all this works. And if you wanna go do this yourself, you're gonna quickly run into this problem. Well, how do I know what sequences to order in my OPAL or whatever if I wanna start doing RNA SABER for spatial transcriptomics? So a while ago, we introduced a bioinformatic pipeline wrapped around a utility called Oligo-array to do genome scale probe mining for the original Oligopaint work. It worked reasonably well. It was very slow. I'm going to skip through the details there. The problem is an interesting problem. So we scan through the letters of the genome or the transcriptome, and what we're looking to do is kind of highlight the stretches of sequence that are the right length that we think are going to make good sites for hybridization. So these would be sites that are effective in terms of the thermodynamics and hopefully are specific, so that it's going to bring the signal where we want it, but not blow up lots of background sites. A couple of years ago, with some colleagues, I introduced a tool called Oligominer, which is a Python framework for designing these probes. It's published and on GitHub, so I'm going to skip these details so I can take more questions at the end. This essentially made things a lot faster, is one of the most important take-homes. But the slide I want to leave you with, and this is a bit of a teaser, but if you're interested in this, the good news is that this got a whole lot easier really recently, and we're actually, it's about to get a whole lot easier. So the work is done, and then this has really been the first project in the lab we're going to put out that's been led by Elliot Hirschberg, who's a research assistant in the lab. And what Elliot has done is put together a really fantastic computational resource called PaintChop. And what PaintChop is going to do is essentially let you interact with a web application where you tell it either I wanted to target this transcript or these transcripts, or I want to target this set of DNA, this region of DNA or set of regions. And the web app can pretty much handle everything. So it does n-dimensional set design. So if you want to target 100 different transcripts and do something ambitious, it simplifies all that for you. All you need to know are the RefSeq IDs of what you want to target. We automatically take things like strandedness of the target, if it's an RNA, into account to make sure you get the right oligo. You're actually targeting the RNA instead of synthesizing a probe with the same strand as the RNA, which would miss it. We've loaded a whole lot of pre-existing sequence collections on there, including some of our own pre-designed probes, as well as a brand new set of genome-wide oligos that we've designed that we're pretty excited about, that we think are kind of the cutting edge in terms of touching on the efficiency of hybridization and specificity points I mentioned briefly. And this is in part all powered by some really nice machine learning that Elliot did to help try to predict the specificity in a scalable way, which is a very tricky computational problem. So this is going to be going online hopefully next week on bioRxiv. So if you're interested, keep an eye out. It's about to hit the world. And I think this is really going to make getting set up with these kind of experiments a whole lot easier. So I'm excited to see people start using it. I'm going to stop here. I hope I didn't run over too much. I'd love to be able to take some questions if there are some. And I'll just quickly acknowledge the people in my lab and the people that help with the work, both at UW and from my time in Ting's lab and Peng's lab, and the funding sources at the bottom. And I'm going to stop here and take any questions.
Yeah, this is a terrific talk. I think if anyone has question, you can either unmute yourself. I believe you are able to, and go to ask, or you can use the chat box to ask questions. So maybe I can get us started with well, 2 questions I have. So I think the PR very impressive. I think how simple, how elegant it is. I love it. And so I think I used to do a lot of MDA, which is similar to RCA. So we try to do sequencing. My lab never been good at imaging, but we noticed I think MDA or RCA, the problem for sequencing is the amplification is not really linear, so we don't get a very quantitative so quantification if we are looking at transcript expression level. So in your case, maybe that may not matter because if you use super resolution imaging, you'll basically just quantify single molecules. But I still wonder whether or not you see your approach does give kind of more linear and a more quantitative, consistent amplification.
Yeah, that's a good question. I guess I'll just repeat the question succinctly since this is recorded. I don't have to repeat the whole thing. But the question is, does PER, and I guess by extension SABER, provide linear amplification of the signal, if I'm following correctly? Yes and no is the short answer on this. You kind of touched upon. So for segmenting objects and saying I can see a dot, I'm just going to count the number I can see. The amplification doesn't necessarily have to be linear to deliver that. And we definitely see that tends to, our ability to do that seems to be pretty good across different amplification levels as evaluated by extending the concatemer different amounts and trying to generate more fluorescent signal. So this is something that's in the paper if people are interested. I didn't go into it here for time. You can actually make like branch structures. You can do multiple rounds of iterative PER where you recognize a concatemer with a sequence that then seeds another concatemer. And Sinan might have touched upon this. So at some point when you start to really go nuts with this, you'll blow out the diffraction limited signals and make them bigger. And at that point, you've pretty much lost, I think, the ability to do quantitative analysis on individual dots. But in general, what we see over like what I call linear PER, so just doing one round of concatemerization with one sequence, we tend to see diffraction limited objects stay diffraction limited. It's a little trickier to evaluate how the fluorescence changes as a function of amplification level. Interestingly, I think because what we're reading out is multiple things at once. So as we extend the concatemer longer, what we generally see is an increase in signal to noise or absolute signal in our puncta, and then it kind of levels off and maybe drops a bit. So I think what you're seeing as it plateaus and drops is probably a loss of efficiency of either docking the fluorescent oligos and or getting the the extended probe just in there. So you're kind of simultaneously reading out a lot of performance aspects that go beyond just the fluorescent signal intensity at that point, if that makes sense. Rong, you're muted. You said you had a second question?
Okay, maybe I'll let the audience ask a question first. Seems like one question in the chat box.
Okay, so Dean asks, what is the smallest number of oligos I've tried with SABER? On the DNA side, we have not seen, I don't think we've seen great success with anything lower than the current kind of state of the art for Oligopaints in general, which is a couple kilobases at best. So that would be 50, maybe 40, 50 oligos minimum. The issue with trying to get really small target DNA fish in general is not a signal issue, it's a hybridization efficiency issue, unfortunately. And for some reason, even if you have 50 oligo probes, we only think that 5 or 10 of them bind. We don't know if it's the same ones or why that is, but that does seem to be the reality based on single molecule experiments. So we basically just can't amplify what's not there. So that bounds us on the DNA side. On the RNA side, we've used about a dozen, and that seems to work. I personally haven't tried to push it lower, but the efficiency there is much better. So in theory, it probably could be done. Like Rob Singer's lab used 5 heavily labeled RNAs to look at the actin molecules, probably single molecule-ish at least, and that seemed to work. So I'll take the next question from chat. So he asks, for the blinking experiments, when you record for a long time, how do you rent photo bleaching of the floors? Good question. So in this case, because the fluorescent oligo is freely diffusing in solution and then just transiently colliding with the strand that we've anchored at the site of imaging, We basically don't. But because there's such a large population in solution, we effectively never bleach out what's available. Even if we bleach it when it binds, that's such a small amount of what's available that is essentially negligible. This is only, but the caveat there, of course, is that this only works if we are using selective illumination. If we're like blasting the whole sample with a huge column of like highly intense laser, then like for sure we could just bleach the whole thing out. But it's a little bit different than a storm approach where you've anchored a dye in place and you're asking photophysical properties to allow the blinking to happen. Once that dye blinks out, in the absence of doing something like the probe refresh storm from Josh Vaughn's lab, you're pretty much done at that point.
Okay, I see. So in this case, probably you have to use turf to image, right? Because you have so many fluorophores in a solution on your image.
Yeah. So we typically for stuff in cells we'd be using high-low or dirty turf, and you could also get away with other geometries. So the image I showed you actually came off a spinning disk. That was a fun wallpaper. I didn't go into details there, but so there we're not really getting 100% selective illumination, more selective collection. But in any case, photo bleaching didn't seem to be greatly affecting the blinking rates we were seeing. So you must not have been using too much intensity. So when you do a DNA SABER, have you tried whether or not you can resolve like a single point mutation in DNA? Yeah, that's a great question. No, we have not yet tried to resolve something that fine. So we know in theory we can differentially hybridize a FISH probe that targets that a single base change in a competitive hybridization experiment where the probe that has not the change is also present. That's really important. Without the competitive hybridization, it doesn't work. But we typically do this on groups of hundreds or more on the DNA level. It'd be interesting to see if SABER could be used to read out. We're kind of kicking around some ideas that might try to bridge what people have done with molecular inversion probes and some of the other probes that would have the specificity to like ligate over a SNP or something like that were more traditionally amplified than with RCA. Maybe SABER could have a place there as the readout, but in terms of, I still struggle a little bit with the hybridization efficiency problem. I think even on the RNA level, that's tricky. I know Arjun's lab and Alex Van Nunaren's lab has published RNA FISH targeting single variants and transcripts, but it still seems tremendously hard to me. It's a very small thing to see on the probe. Yeah, I think I have another question or comment. I think that's the experience I had, I think, early days, about 15 years ago, when I started as postdoc. I think I was trying to design the DNA probes to look at different transcripts and also the genomic loci. And that time, I think DNA microarray was still in a state of NAR. I think the Affymetrix did so much work to validate their probe to do really genome-wide gene expression profiling. So they have so much data there. I don't know if they can share. We got a list from Affymetrix that time. That helped me a lot because we tried to do computational probe design. I think about 5 probes is so difficult to look at the probe cross-reactivity, but they give us a list of 2,000 probes. That helped me a lot. Okay, that's great. Maybe you can reach out to them. So now they are part of Thermo Fisher now. Okay, that'd be interesting. Yeah, and I think if it ever comes to it again, we do have pretty scalable ways to ask that question now, but it's only because of the machine learning we've done. Like so much computational. Yeah, it's a huge part of how this all works, but it's sometimes the less flashy thing to talk about. So, well, if people are interested in that, there'll be a discussion of that in the Paint Shop manuscript that's going to go on bioRxiv probably next week. Mm-hmm. All right, so any more questions from the audience? Okay, if not, let's thank Brian again. This is a really terrific talk. Yeah, thank you. Yeah, thank you very much for the invitation. It was a lot of fun. Oh, you see people applause. Okay, thank you. You guys have a good weekend.